In this video, we are going to talk about DNA replication. What I've done here is I've combined the SL little subtopics here, objectives, with the HL, and we're going to kind of glance over these kind of quick. So that's why they're kind of mixed up here on this slide I just kind of invented. But before we do that, let's talk about why. Let's talk about why we need to copy our DNA. So it really comes down to just part of the cell theory, right? Like every cell, this is a cell and this is the nucleus. Every cell comes from pre-existing cells. So in your body, through the process of mitosis, you end up with two genetically identical, and that's the important two words, genetically identical cells. When I say genetically identical, I mean that this nucleus and this nucleus have the exact same chromosomes, the exact same base pairs, everything exactly the same, no matter what cell it is in your body, because remember, you have 23 pairs of chromosomes, all with the same genes in every single cell, whether it's a muscle cell or whether it's a nerve cell or a cell in your retina. That's also a nerve cell. But anyway, so they're all identical. So the important thing is that I have to take a double helix of DNA that I have in this nucleus here. I have to take this and I have to make a copy of it, and I have to put each one of those copies into the cell. What we're going to look at is how this works is we actually split your DNA and we copy a new strand from it. So we have, actually, let me split them first. So we have, we've split this into two like this, and then we just copy them. We just read the bases off, and when we're done, we're going to end up with an old red strand and a new blue strand of DNA, and each one of these are going to end up in a different nucleus. So that's the reason for DNA replication, is to copy our DNA for mitosis. And we see this in cell division. We also see this in asexual reproduction. Unfortunately, we also see this in like tumor genesis and when cells are dividing uncontrollably. So that's what we're going to look at. We are not going to spend any time on the SL stuff because the HL material talks about this and then some. What I do want to talk about in the SL category here, list of the SL topics, is this idea of complementary bases, complementary base pairs, conserving the DNA sequence. So what does that mean? So complementary bases, if you remember, is that A, adenine, will always form a bond with thymine, and that cytosine will always form a bond with guanine. And because we have that, we are guaranteed to conserve or to save the DNA sequence. So you might be like, well, that doesn't make any sense. So let's just illustrate that real quick. So if I have DNA, I just want to say one thing. There's so many different ways that we draw a DNA molecule. So you just saw me draw it as a double helix, and now I'm kind of drawing it as a straight line. I'm separating them out now, like a broken ladder. But you're going to see many different drawings. So sometimes you might see them demonstrated this way, like a ladder, and then them just connected like this. So that's what I've done over on the right-hand side here, but I've split it down the middle. The other thing I want to say is I want you to visualize that this here is a nucleotide, and it looks like a T on its side, right? It looks like this, right? A T on its side, but think of it all the time as our phosphate connected to our sugar, connected to our base. So anytime you see this or this part of the DNA, I want you to think nucleotide. Okay, so what it means by conserving the base sequence is that because let's say adenine was here, that means on this other one it was thymine, right? And because let's say cytosine was here and guanine was here, that means, oops, my little G. That means on this side we had thymine, correct? We had cytosine. I did that backwards. We had guanine, and then down here we have cytosine. Let's do one more with thymine down here, and here we have adenine. So when they were joined together, they were this way. But I need to make copies of them both, right? I need to make copies. So when I make a copy of this other side for it to eventually end up in a nucleus, I'm going to erase this for a second. Remember, it was TGCA, right? The new nucleotides that are floating around free in your nucleus, the new nucleotides are just going to form pairs here. So there's a new one, and right, we have a little T. Here's a new one, guanine, bound to cytosine. Here's a cytosine bound to guanine. And here's an adenine bound to that thymine. So that's what we mean. Because they complementary base pair, because T always binds with A and C with G, we are guaranteed that the new piece of DNA will be just like the old one that we separated and removed. So that's what they mean by that. Okay. So let's get on to a couple things that sometimes creates a little bit of confusion. So here's an HL statement that says state. Okay, just remember that. We have to remember that DNA replication happens in the 5' to 3' direction. So what does that really mean? So if you recall that our nucleotide, if we look at a nucleotide, we have a phosphate connected to a deoxyribose connected to our base. And carbon number here, right? Remember this carbon here was carbon number 5. And here's carbon 4. And here's carbon 3, right? We start here. Oxygen, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So that means right here we have carbon 3. When we add a new base, so let's say over this side, we have our original piece of DNA. I'm being lazy and just drawing the latter part. I'll draw a little. Let's pretend this is thymine and this is adenine. So as we add a new nucleotide, as we're copying this, and let's say down here is cytosine, it means that a nucleotide containing guanine will connect to this cytosine. So that will form our complementary base pairs, conserving the sequence. However, the important piece is that this will be added on from the bottom, right? And this phosphate will form a bond with carbon number 3. We talked about this in topic 2.6. So that means we are starting at the 5' end, the 5 carbon end, and we're working in this direction to the 3'. We got a 5' and the 3', 5', 3'. So we're constantly moving in this direction when we're replicating our DNA from 5' to 3'.